All right, I'm going to give a slightly different viewpoint here. I'm surprised that I haven't heard this yet this meeting. I represent a different community, well, a biological time series community. I'm Todd O'Brien from the COPEPOD project, co-chair of IGMETS and analysis time series lead for multiple ACEs and SCORE time series working groups. And I'm going to give you a brief intro to what we did to address data sharing challenges, and then during the workshop, I'll go into the tools themselves. Do it on old screen. Okay. So time series data are different from other data. Other data get less relevant each year. An individual observation without temporal before and after measurements is what I mean by other data. They're arguably less relevant after 10 years, and after 30 years, they may be obsolete. Why? Well, in a changing world ocean, what the ocean was doing 10 years ago or even 30 years ago doesn't represent what it is today. I've seen that in the zooplankton biomass, where areas have crashed, and if you look at 30-year-old data, it's much higher than it is nowadays. Likewise, methods and gears change, unless you are purposefully trying to keep them consistent. But if you're trying to use the latest and newest info, you're not going to keep it for 30 years. Time series, however, which are repeat observations to create a temporal change of before and after states, they start getting really interesting at 10 years, and they are super valuable at 30 years. So unlike other data, where every year it's older and older, and you may not care about it as much, time series, every year your time series is longer, it becomes more valuable, and you may become more protective of it. So yes, time series data are valuable. Here's a simple chart, the scientific value of time series, and by that I mean what types of questions it can answer. On the y-axis, we have increasing value, and on the x-axis, we have less on the left and more on the right. So let's look at increasing time series length. The longer it is, the more valuable it is, the more questions it can answer. At 3 years, you have a good idea of the seasonal cycle. At 10 years, you can start pulling out statistical trends, especially in loud biological data. And at 30 years, you can start pulling out the climate change signal or climate signals. If you look at the sampling frequency, more frequent makes it more valuable. Once per year can give you a noisy interannual signal. If you do seasonal sampling, you get a better picture. If you go to monthly sampling, you can see the seasonal cycle and how it changes between months. And nearshore spots, you can get weekly or sub-weekly, and in estuaries, you might even go hourly. So more frequent sampling makes the data more useful for more questions that you can answer with it. What about co-sampled variables? Sampling a variable just by itself has value, but if you add additional variables, temperature, salinity, nutrients, and other things in the environment, it lets you tell more about what's going on and explain why. If you only have zooplankton biomass and they crash without temperature, salinity, nutrients, chlorophyll, you can't really say what happened. And here, how about the critter time series? By that, I mean fish, plankton, phytoplankton. The resolution that you measure your data at increases the value and the things it can answer. The most basic is total biomass, where you just measure the entire volume or biomass of the sample you took out of the net. If you run that through a sieve, you can have size fraction biomass. Somewhere in here, you have the optical instruments that can measure size and identify two groups. And taxa groupings are useful. Is it a COPEPOD? Is it a gelatinous? But you want to look at biodiversity and exactly what's going on. You really need full speciations. That's, counting with a microscope. Counts are probably the easiest. But if you can also take individual biomass, you expand beyond that with what you can do with these data. work, and carbon and ecosystem balance and modeling work. So why some time series will not freely share their data? As I said earlier, I'm speaking on behalf of multiple Plankton time series groups happening over the last 20 years. We have over 150 ship-based Plankton time series, and if you add ship-based biogeochemical, that gets up to over 350 time series. We did not include temperature-only time series in our collection. If you add those, it's even higher. In a world where funding is 3 to 4 years, and you're trying to do a 10 or 30-year time series and be unique and change something new and exciting each time, it adds a challenge. So many Plankton time series PIs simply want the connection of a direct data request so that they can offer friendly advice and guidance on the people using their data and to keep track of usage so that at the end of the year, they can tell their boss, this is who's using our data, this is why it's important, these are all the papers published from it. Plankton data has huge sampling differences depending on the type of mesh you use, the type of net you use, your counting methods, and the taxonomic resolution that you use. An example, I'll go with the CPR data. You could say the CPR data has phytoplankton, but you need to add that it's a 270 micrometer mesh net. The phytoplankton is being snagged in there. It's not really being caught. It's a shallow, high-speed tow device, and it obliterates large zooplankton, large gelatinous and large zooplankton avoidant. So how you capture that in the metadata record and get people to read that and apply it when they're looking at the data is a challenge. So here's another plot. On the y-axis, we have likelihood of sharing data. And on the x-axis, we have less to the left and more to the right. So step one, how much time or how easy is it to generate a cruise worth of data? Is it automated? Can you get your cruise done in 3 hours of running it through scripts? Or does it require lab work? Does it require a lot of lab work? Or does it take you weeks and weeks of microscope time to count every single little critter in the sample? The more effort you have to put into it, the more unlikely it is people will freely share it. And if there's an embargo of only a year, they may not have even processed their samples. Someone who got their data done in 4 hours had a whole year to look at it, and they may finish their data the month before. Do they really want to give it up? How about expertise? I'm pretty sure we could give a thermometer to anyone in this room, and they would be able to measure temperature and understand what it meant. You could probably give the average undergraduate or graduate student simple CTD or calorimeter, and they could probably get it to work. But when you start needing trained technicians and chemists to do it, it's more work. It requires more expertise and trained staff at the time series program. And if you want to look at biodiversity and all species, you need trained taxonomists that are familiar with an area for years and years and years and years. So that reduces the likelihood of someone sharing full species plankton data versus CTD data. Similar thing, how complex is it to understand the value you get? Temperature is pretty basic. Taxonomy and some of the biogeochemical things are fairly specialized. This is a big one. Availability of funding. If you look at the world and places that share their data the quickest, most of them have secured funding. Australia is really incredible. NOAA is pretty decent. In reverse, the hardest people to get data from are institutes where people are competing with each other violently. I've had cases, here, Todd, here's my data, don't give it to this person at my own institute. And, you know, that's just crazy. And I've had cases where I have the Plankton data, but I can't get the CTD data. Totally reverse of what I would expect. Solution, more funding. Solution, more staff. If they can get the data processed quicker and have more time to analyze it, they will be less protective of it and more likely to share. So when we talk about FAIR and metadata and all this, how likely are these people to give their data? When you ask them, if you go, hey, I would love your data to add it to this, I'll take whatever you have in whatever format you have, and I'll ask you questions as I do it. Can I please have it? I'd really love to work with you. That's fine. But if I go, you must use Darwin ISO 666 XML, and you must provide it in this format, that really changes their willingness to do it. You're Plankton taxonomist and you look at raw XML, it spins your mind. They don't want to do it. They don't have the expertise to do it. This was true years ago. I'm sure it's true today. Years ago, if you went to a grad school, the physical oceanographers all had workstations on their desk. The chemists all had PCs on their desk. And the biologists, unless they were modeling, had a Mac and they use it for email. I'm sure that's changed now with more modeling and optical stuff, but the computer level drops as you get into the biological data. In a publish or perish merit system, any time you spend making your data fair is potentially time spent not writing your next paper or grant proposal. And as Justin said, unless, as the discussion said, unless the funding agency or journal require the data to be public, it's a distraction. And that's the biggest change has happened since these requirements came about. So, what am I doing for time? Two quick examples. Again, I'll talk about these in the afternoon session. IGMETS is the international group for Marine Ecological Time Series, a group that I co-chaired. And PLET is a Plymouth Marine Laboratory. Claire also led and mentioned the Plankton life form extraction tool. And these are two examples of creating cooperative shared data products based on data from sometimes uncooperative and non-sharing communities. IGMETS was working with Biological and Plankton Time Series. We published a status report with a huge annex listing every single time series that participated. It's about 150 pages just by itself for the annex. We created a time series explorer, which I will demo later. That's on the left, and a metabase, an interactive map that you can click on to get time series information and contact information. We ran this with a single data processing person, myself. We promised we would only use the data for the analysis in the report, that we would not share it or release it without their position and permission. Instead, we would direct people, go talk to Justin if you want his data. He can give you the newest data. That's also important because we requested aggregated data, not the raw species. We worked with total COPEPODs, total diatoms, not species level data. So you have to go to Justin or whoever to get the full data. We requested 2-year-old data, which by the end of the project was 4-year-old data. So at that point, the countries that were still shared are nervous to share with us, let us use their data and participate in. And the participants were co-authors on the regional chapters within the report and featured in the annex and in the interactive metabase. But again, we did not share any raw data. We just provided information on how to request it and who to contact. The Plankton life form extraction tool is actually really interesting. And if I did IGMETS again today, I would probably follow their model. It started in the North Atlantic, but it appears to be semi-global now. It contains raw species data, and you can download some raw species data, but the individual data sets have different access levels. Some are public, some are restricted. The raw data, you can only get the non-restricted public, but it creates aggregated products, and there the restricted ones participate because they're calculated into something new, not just raw data. So I'd already said that. The aggregated products are useful groupings commonly used in status summaries and ecosystem overviews, stuff like microplankton, diatoms, small copepods, large copepods, harmful algae species, and there's many more. It's a higher level of looking at the data that's easier for many people to understand. Therefore, it's more useful to when working with policymakers or writing more basic reports. And again, everything can participate in the aggregated, and you can download it. But the public stuff, you can also get raw species versions of it. And I think that's it. So again, I'm coming from the plankton community. I hear people say they put their data on right away, but based on the microscope time and the expertise and the shortage of taxonomists and the challenges of the data, we have a different challenge than the chemical and physical groups. Thank you.